All right, good morning, everyone. So let's get started with today's class. Just a reminder, if you're online, please make sure that you turn on your cameras. Let me share my screen here and let's get started. Okay, so last class, we were talking about some experimentation concepts. As a reminder, this presentation is on our website. So it would be on the lectures for last class period from January 25th. So if you want to follow along, it's going to be there. I also have another one posted, which is what we're going to be following up today. So if you're following, first let's finish the one from the January 25th, and then we'll get on today's presentation and go from there. All right, so the one that we were talking about last week was this one, experimentation concepts. We already went through most of this presentation, so my goal here is just for us to quickly do an overview of what we talked already about. These concepts are really important for you to know and be aware of. I do hope that you already are coming with some of these concepts from previous stats courses, but if not, I do want to provide you a solid foundation because these concepts are going to carry through every statistical analysis that you do that's related to experimentation or plan design studies, right? So these concepts are very important that you understand, not just for this course, but for your own analysis as well. All right, so I'm going to go very quickly through the slides that we have already covered. I'm not going to spend a lot of time there, just as a refresher of what we talked already about. So in this presentation, we're talking about an experimentation planning workflow, hypothesis testing, a study design that's made up of treatment and experimental design. This is, again, a very important distinction because they do have different purposes, as we're going to see in more detail today. This concept of experimental unit versus observational unit, another very important concept because this is going to determine if you have true replication or if you're doing subsampling, which impacts on the model you run, on the standard errors you get and the p-values you get. So basically impacting all of your analysis results. And then we're going to talk about a couple of experimental design examples, like the completely randomized design, one of the simplest designs that we have in applied sciences, and also the randomized complete block design, which is a little bit more complex and helps us to address extra sources of variability. Okay, so we're just saying, what was a workflow, an experimentation workflow that would basically resumes, I guess, or is summarized in this figure where we have the treatment design that's going to be derived from your curiosity, your objectives, your hypothesis. So really treatment design is addressing your questions, right? I want to test this, so I will have these treatments to help me answer that question. Whereas experimental design is the part of the design that is related to experimental material homogeneity or lack of homogeneity. And that's what helps us to address some of those components and remove that heterogeneity from the effect of the treatments and be able to still find treatment effects. We talked about a good way to set hypothesis. Remember, hypothesis should be testable. If you have a word in your hypothesis that makes it be true, whether you find differences or not, that's not a good hypothesis. You have to be bold, you have to make an assumption and write it down in a way that your hypothesis is testable with your data. So we ran through some examples. I'm just going to skip through those. And of course, your objectives should stem from your hypothesis. A lot of times people don't have. So if you have a presentation and you're not showing neither hypothesis nor objective, then you need to revisit that presentation. I was in this seminar once that there was a student presenting just like an overall seminar on their research, and they did not present hypothesis or objectives. So they got all the way to the end of the presentation, and you're looking at that and you're like, okay, so what are you trying to answer here? What is your main question? If you don't have that, you don't know. So one person in the audience raised a hand and said, can you go back to your objective slide? And the student was like searching for one and they didn't have it. They're like, well, I don't have one. It's like, well, that's the point. You should have one in every presentation, just to, for it to be clear to you what you're doing, but also to your audience as well. So the study design is really comprised of these two pieces, the treatment design, experimental design. The treatment design is the component of the study design, again, that addresses your objectives and hypothesis. So you should be choosing treatments that when you compare them, you should be able to answer your objectives. So we have this example here where we're talking about using different nitrogen and potassium fertilizer rates to optimize yield in a given crop. I think we're talking about corn here in this example. So it would select multiple levels of those factors, right? So another distinction here is nitrogen fertilizer and potassium fertilizer are treatment factors. Now you can kind of think of that as like the overall hierarchy of your treatment design. And then under your factors, you have levels, right? So sometimes that distinction is not very clear. I just wanted to make that distinction clear here. And then because we're interested in the interactions of these two factors, we also need to have all those 3x3 combinations between nitrogen rates and potassium rates, so we can assess the main effects. So the main effect on nitrogen, the main effect of potassium, but also if they interact, and if they do, how so? So we need to have all those treatment combinations to do that. Okay, so again, the treatment design is addressing your objectives. Now, what is the use of the experimental design? So this is where we're going to understand our experimental material homogeneity. And if it's not homogeneous, then we should use experimental design to address that lack of homogeneity. Right, and not just homogeneity, but also limitations. So if you are working with, well, I guess, there are probably many examples of this, but the one that I can relate more to is if you think of perhaps you're doing a nitrogen by irrigation study. You can probably apply nitrogen to a smaller area than you can apply irrigation treatments because perhaps you're using an irrigation pivot to apply irrigation treatments, which requires a larger area to be treated with a given irrigation level. And that may not be the same as for nitrogen. So perhaps nitrogen you can apply with a tractor and a spreader on a smaller footprint. And so the experimental design is also going to allow us to handle those limitations of space, and especially when different treatment design or different treatment factors are applied at different sizes of experimental unit. So our goal here is to choose the simplest experimental design that will allow us to accommodate all of our experimental units in homogeneous material. Then we got into this concept of experimental unit and observational unit. So again, experimental unit is a really important concept because this is a lot of times is not clear cut. And for your specific study, you should know what is your experimental unit based on what level you're applying your treatments. So if you're applying treatments to the whole plot, in this case, the whole plot would be within those two flags and stakes. That is your experimental unit because that's the level you're applying your treatment. However, sometimes the experimental unit is not the same as of your observational units. So what we do then is we go out in the end of the season, and this example here, collect 6 corn plants to estimate yield, biomass, and other things. So our observation unit here is corn plants, individual corn plants, in this case, a bundle of 6 corn plants, whereas our experimental unit was the whole plot, right? So this is important because if you come here and let's say you do decide to measure yield on a plant by plant basis and you have 6 yield levels, 6 yield data points for this plot, you should not treat them as true replicates because you do not apply treatments to the plants, you apply treatment to the plot. Now you're coming here, you can use still the 6 plants to give you an idea of how yield behaved in this plot, but you should really be thinking of this plot being one observation for you and not 6 like this. And that impacts on the degrees of freedom of your analysis of variance models, which impacts your p-values, pairwise comparisons, everything. So again, this concept here is very key to get it down. Then we talked about a replicate and a block. So a replicate would be in any design. A replicate would be the different experimental units that receive the same treatment. right? Whereas a block perhaps is a very specific concept for randomized complete block designs, where a block is considered a unit in space, where under it you have each treatment appearing at least once. So we have this this drawing here just to drive that point home, where block one is the red rectangle, where we have 9 plots, where each plot was randomly assigned to a treatment. And then, of course, we have the plots as you see the cells inside the block. And then we have the plants here depicted by the green dots that are our observational unit. So we have these different levels of hierarchy in an experimental design. It's important to know as well. Okay, so on the example that we're talking about, we had 3 nitrogen rates, 3 potassium rates. We do all combinations of those 3 levels and get 9 treatments. And let's say I want to have 4 replications. Yes.
I have a question regarding experimental unit, blocks and replicates.
Mhm.
So in replicates, you can randomize the plants by keeping the treatment same, right? And in blocks, you can change the treatments.
So we're gonna, so a replicate exists in most designs. So both on completely randomized design, which is the simplest, and in randomized complete block design, they are replicates, right? It's just that in the case of randomized complete block design, we also refer to a replicate as a block because it's going to contain the entire replicate of those same set of treatments. The main difference, which we're going to see here in a moment, is how randomization is restricted or not, depending on the design, which that connects all the way back to your experimental material being homogeneous or not, and also in the end, how we run the model. Because what could happen is you may say, okay, I need a randomized complete block design because I have different soil types or whatever it is, and you run that on the field. So you do all the pre-experimentation, like planning. So we do a randomization, you do a treatments, do randomization by block and so on. You go out and establish that study, but then once you collect that study and come back to your computer to run your model, there's nothing keeping you from specifying the wrong model except yourself. So it is important for us to really understand, all the way from the planning stage, what is the limitation that we may have, make sure we randomize properly and we choose an experimental design that addresses that. And also after you collect the data, that you come back and you run the proper model that represents what you did on the field, which is based on what you planned beforehand. So we connect everything, right? Yeah, but so I guess getting back to your question, we're going to see an applied example here where I think is going to help you to visualize that. But if not, please make sure to ask again and we can talk about it. Okay, so, so we have those 9 treatments, and let's say we decide to have 9 replications. That means that we need 36 experimental units, right? So now I know how many units I need, and let's say that I am running a field study, and I know how many rows of crop I want to grow within each experimental unit, and what's the length of each individual plot. So this is going to give you an idea of how much area you're going to need out in the farm to have this study, right? So you go to your farm superintendent or maybe to your professor and say, hey, let's run this study here. I need half an acre of homogeneous soil. Then your farm superintendent may come back to you and say, well, I have this field here that I get 3/4 of what you need under one soil type and another 1/4 in another soil type. Does that work for you? If that's the only thing they have, it's going to have to work for you, right? You're going to have to make it work. Of course, if they can find somewhere that's more homogeneous, that's better for you. But if they cannot, which most of the time that's the case, we have limitations in space or funds, or in this case, homogeneous material in a farm, then you may need to address that using your design, right? So the question then that follows is, once you know how many units you need, and in this case, what's the area of those units that you require in the farm, then you need to ask yourself, do I have enough homogeneous experimental material to accommodate all 36 experimental units for this study? And this question here is going to determine which experimental design you use, right? So again, the two most commonly in applied agriculture, two most commonly experimental designs in applied agriculture is completely randomized design. That's the simplest one, and a randomized complete block design. That's perhaps it's a little bit more complex, but it's really one of the most widely used in what we do. So if you have this question here is, if you have homogeneous material to accommodate all those experimental units, and if your answer is yes, then you can use a completely randomized design as the design for that, and treatments will be randomized to the entire area or maybe to the entire experimental material without restrictions in randomization. And this is key to why we do this and how this contrasts to other designs. So just keep that in mind. There are no restrictions in randomization when you're doing a completely randomized design. So here I'm just showing you one example here. So imagine that this is my piece of land at the farm, and the farmer superintendent told me that they were able to accommodate all of my experimental units in a very homogeneous spot. So same soil type, perhaps no elevation gradient, very, very homogeneous soil type is what we can tell. So then we go here and when we randomize our treatments, really, we take each one of these cells that you see on this grid, it's going to have a number, and we're just going to randomize all of our 36 experimental units to any one of these plots in the farm. Meaning that what happens, because we have no restriction in randomization, it could be that maybe a same treatment could fall the same treatment could fall in neighboring plots, right? Because you have no restriction randomization, you're just randomizing any single treatment to any single experimental unit in this case. So I'm just highlighting here the treatment one and its replicates across the field. So what you can realize is that because we had no restriction in randomization, treatment one appeared 2 times in this first column, appeared once in the second column, it appeared 0 times on the third column, and 1 time on the fourth column, right? Because again, we have no restrictions in randomization, so this could happen. And a very similar thing happens with other treatments. So number 3, for example, is you have 3 and 3 right next to each other, and that should be okay. Because since we are assuming that our homogeneous material is, or that our experimental material is homogeneous, then the fact that by random chance, the same treatment fell right next to it in 2 different plots should not have an impact on the analysis when we are estimating treatment means and performing comparisons. Okay, so what if actually you do not have homogeneous experimental material? So your farm superintendent comes back and says, well, sorry, we don't have that. That example I gave earlier, maybe I have 3/4 of this area in one soil type, the other 1/4 is a different soil type. Can you work with that? Well, I guess we can, right? So that's when randomized complete block designs come into place. So this is just an example that is kind of driving this idea of this example. Let's say that I was able to fit 2 blocks in one soil type and other 2 blocks in other soil type. If we just keep our completely randomized design as before, so I'm just showing you here different soil types, but still the same randomization for a completely randomized design. So let's see how many comments here. Yeah, so if we look, at least if we pay attention to treatment number 1, what happens here is that treatment number 1 now is appearing 3 times under a dark soil type in this example and 1 time in a light soil type. If we assume and if we make the connection that a darker soil type may mean that it has more organic matter, more soil and nutrient holding capacity, it's just a better soil overall than a light colored soil that may be sandier, may be leaching, more leaching is happening. So water is draining, nutrients may be draining with it. So we're assuming that dark soil type is better or has a greater impact, positive impact on yield, right? than our light color here. What do you think is going to happen if we just think of treatment number one in this case? How would it, what implications is this randomization in this setup going to have when we compare treatment one with other treatments? Yeah, so more bias, right? So we're saying here, so let's say that treatment one is your zero check. So we apply no nitrogen. Then maybe another treatment that ended up being on this, on the light color, I don't know, maybe we can look here, maybe number 8, we have 1, 2, 3 number 8s on this column here. So let's say the number 8 is your 100 kilograms of nitrogen per hectare, and number 1 is your 0 nitrogen per hectare. Now your your treatment number 1 is gonna look a lot better than what it should maybe, than your 100 kilograms of nitrogen per hectare, just because of soil type differences. So it has nothing to do with your actual treatment effect, or it has some to do, right? But that some to do is confounded. You cannot separate your treatment and your soil type effect. They're combining to that average that is biasing your treatment effects in this case. So if you run this study here, you're probably going to have some very large standard errors, which is going to make it difficult for you to find treatment differences in this type of setup. Yeah, so I guess here I'm just saying that again, that in this case, treatment number 1 is going to have an average yield that's overestimated because it appears more times in the soil type that's good and less in the one that's bad. And also because the way that we randomized these treatments to the experimental units, which was without any restrictions, it is impossible to separate that effect. So now the treatment means, or the treatment observations and afterwards their means, are going to have that effect of the treatment, but also the soil type. You cannot separate them because of how they were randomized. And so what ends up happening is we're going to have a negative effect on both the treatment means, right? So they're going to be biased, but also this is going to inflate your standard error of the analysis. And really, a standard error of an analysis of variance is going to drive the p-values of our ANOVA table. It's going to drive the pairwise comparisons you make on the treatment means. So it's really going to impact the whole analysis if we make a decision like in this example here. However, there is a solution for that, right? So randomized complete block design would be this experimental design that we could use here to overcome that issue. So what we would do then is we would have blocks that would be completely confined within each one of those soil types in our field. And just a recap here. So a randomized complete block design, what do we mean by that? So we basically mean a design where each treatment appears once per block. So that's one way of thinking of it. The other way of thinking of it is that each block has all treatments appearing once, right? So if you have blocks that don't have some treatments, it's going to be a different type of design. It's going to be an incomplete block design. And that is an actual true design that exists that people use. I don't think it's very common, so we're not going to cover that in this class. If you have more complex designs, you will need to take other classes. But here we're really talking about these two most common ones, and we're going to focus on those. Okay, so now I'm showing you a picture here of how a randomized complete block design for our previous situation of heterogeneous experimental material, in this case soil type, would be. So notice now that I am labeling these columns as block 1, 2, 3, and 4 in a way that they are completely confined within that source of heterogeneity, in this case, soil type. Also, what you may notice now, let me just run here, is that each treatment is appearing once on each one of the blocks. And this, again, is by design. This is what this study does. And the way that we accomplish that is by restricting randomization. So on the completely randomized design example, imagine that you have, I guess, before computers, people would use hats and would draw treatments into plots, right? So let's say that this is plot 101, 2, 3, 4, 5. So we have 101 through 109, 201 through 209, and so on. In a completely randomized design, you would have, if you're drawing from a hat, you basically have one paper, scrap or scrape or one little paper piece with each one of your treatments. So let's say you would have, four paper pieces with number 1, 4 with number 2, 4 with number 3, and so on. And then you would come and say, okay, let me pick a treatment for the first plot, 101. So it would go there, take, and it could be, in this case, it would be number 6, right? And, or maybe not in this case, we're talking about the randomized complete block design. So it would be on this case here. So you took the first paper with number 2 from a hat, and then you assign that treatment to that plot 101. And then you do again, and then you say, now this next one's going to go on plot 102, and then it's number 5. And you keep doing that without any restriction, just following that sequence of plot number. In RCBD is going to be different, because now for each one of these blocks, you're going to have one hat with each treatment appearing once. So I'm going to say, okay, for block 1, plot 101, you pull a number, that's number 6. And the number 6 is not going to be pulled again for this block because it's already selected, where you only had 9 pieces of paper that you're drawing from this hat. So you complete all the randomization first for block 1. Once you finish pulling all those 9 treatments, all those 9 pieces of papers, you put it back in your hat, you mix it again, and now you do again the same process for block 2. So now your randomization is restricted by the block. You're randomizing first one block, then the other, then the other, then the other, in this way that each treatment appears once per block, and every block has each treatment once, as we're seeing here. As I was mentioning, these are the 2 of the most common designs that we have in agriculture, but they're not the only ones, right? And so other designs that you may have heard, maybe you are using in your research, some examples would be Latin square, where instead of having one blocking factor, like our blocking factor is soil type, maybe you have soil type and elevation gradient. You would have two blocking factors that can be fit into a Latin square. There are balanced incomplete blocks, partially confounded factorial, cyclic, lattice, supersaturated, response service, and the list goes on and on. There's a whole course just on experimental design where you can learn about all of these. And again, we're not going to be covering them in this class, but I do want you to at least know that they're out there in case you ever need. There are classes, you can Google it. There's ChatGPT, there are books that you can learn from. But really the most commonly used are CRD and RCBD, and that's why we're covering them in more detail. Okay, so just a quick summary of this lecture here is that, it all starts with curiosity, and perhaps a lot of times what happens, and maybe your case, is it's not like you get to a program, to a lab, and then you tell your advisor, this is my idea, this is what I want to do, let's write a proposal. So normally students don't have the chance of seeing the process from the beginning, right? When you get to your advisor, they already have a funded project, which means they already did the curiosity part, literature review, they already selected treatments, and potentially they selected experimental design already. But it all starts there. And again, treatment design addresses your research questions. Experimental design is going to address lack of homogeneity in or limitations of the experimental material. And the proper choice of that experimental design will allow for you to conduct unbiased and accurate treatment comparisons. All right, so this is what I had for this lecture. Before we move on, let me just take one step here and ask, does anyone have a question? Okay, I don't hear anything. So let's, so I have a quiz for now to just go through some of the concepts that we went through this lecture here. So if you go to your EGA ELC, I'm gonna make this available right now. So this is called quiz number 2, experimentation concepts. Really the idea is that you do this without looking at your material. It's more of if I just want to see how were you following along, and maybe if I was unclear in some points. So please make sure and take this quiz now. And if someone can just confirm to me that that's available on your ELC. Okay,
Yes.
okay, awesome. So I'll give you here 5 minutes. Yes.
Since there are already more users.
Did it work? Okay.
Hi, Dr. Bastos.
Yes.
I am unable to find the link for the quiz.
Okay, so on ELC, let me ask someone, because maybe the way I see it is different from you.
Okay.
Let's see here.
Yeah, the tools, in tools, there's quizzes.
Okay, so if you go, so if you go to tools and you click on quizzes, it should be there.
Okay. Thank
Were you able to find it?
you. Yeah.
Okay, awesome. All right, so our time is done. Does anyone still need more time? All right, I'm gonna take that as a no, and I'm gonna keep moving. Make sure you submit now or within the next 5 seconds or 10 seconds. Because I'm gonna close it. Or maybe I should, let me see, maybe I can leave it open. All right, I'm about to close the quiz. So make sure you have already submitted yours. All right, quiz is closed. Let me see if there's an easy way for me to go through. Our answers here so we can talk about them as well. Okay, so, I hope that I told this enough times today and yesterday or last class that you got this right. But let's just go through this again. So treatment design is going to be related to your objectives, right? That's what treatment design is there for. In your experimental design is the part of the study that is going to allow you to assign your treatments to homogeneous experimental units and those types of limitations. Okay, so then we have this question here on treatment factors and treatment levels. So in this case here, the treatment factor is planting date, right? And treatment levels are levels under that factor. So that would be the May 1st, May 15th, and June 1st. The next, or did I skip one here? Okay, so yeah, sorry, I guess I skipped number one. So number one is our hypothesis objectives should directly affect the treatment design. And then second question was related to that. We already covered it. Question three, we just talked about it. Question four is the two experimental designs discussed in class for CRD and RCBD. That is true, right? That's what we discussed. Question five, experimental design to be used when experimental material is homogeneous. That's completely randomized design, as we just talked about. And then finally, if you have heterogeneous experimental material, you should use a randomized complete block design, at least as far as what we talked about in class, right? So this would be the rubric. If you got a grade, if you know, based on your answers and based on what I'm telling you is the rubric, if you got a grade that you were not expecting, let me know. Sometimes I may have made some mistakes when I was setting up the quiz, but I'm assuming, that it was properly set up. All right, so let's go to our next topic here. So, yes.
Quick question.
Yeah.
So if the RC, like, I would like to make your experiment, I mean, when you do the layout for experiments, it should be parallel to the effect.
Yeah, so for Elisa's question is when you're doing an RCBD, let's just go back to that figure there. Right, so this figure here. So when we have a factor that we know is causing heterogeneity in our experimental material, we should use blocks, right? So your question is if the blocks should be, in this case, the columns or in a row setup, right? Yeah, so the blocks, well, let me ask you that. What do you think? So it would be in the columns, right? Okay, so, yeah, so it should be in the columns, right? But let's just think about it, because really, perhaps one of the worst designs that you could have is actually have known experimental material variability or heterogeneity in this case, and then do your blocks, but do them in a row style rather than a column. That would kind of be one of the worst decisions for this specific case. But why is that? So imagine that now, instead of having our blocks as columns, we actually just, so we kept the soil types as they are, but we just moved the blocks from being in the rows to being, or sorry, from being on the columns to being in the as rows. What would happen then? Right, so block would be within the block, you would have heterogeneity, right? And now, again, we're back to the same place of now you cannot separate the effect of soil type from treatment. We're gonna see an applied example of this in practice, which is, I hope is really, if it's still confusing for you, I think it's gonna be very clear cut, because you're gonna be looking at data, analyzing that data, and seeing the effects on the actual results. But yeah, so we do want to have our blocks completely, so a given block should be completely within that level of variability that you have in the field, right?
Okay, one small question,
Mhm.
little bit complex.
Okay.
Like, most of the times you don't know what's in the field, like, how it is. So you did like blocks like this, like,
Mhm.
opposite way. Like, is there any way to, like, can we back calculate or using the modeling, like if the soil is this much or not, or it's like gone steady?
Okay, so Aminder is saying, is asking, so if you do have variability in your field, but you block in the wrong direction.
Yeah, like half it, like treatments in this and half in the other.
Okay, so then the question is, can we account for that? So there is a way that we can account for that, is to use a covariate. So it would be more like analysis of covariance, where you would still have your model, like your model will still be the one that integrates your treatment design and your experimental design, but then you may add extra variables in that model. So let's say, in a case, if you think of this case here, if you do have your blocks in the row direction, you may add in a variable. So imagine your data set, you have a column saying what is like so dark or light soil type. So maybe you can add soil type here. Perhaps if you have a numerical variable, you could, like elevation, for example, you could add that average elevation of the plot as one of the covariates. It is a solution and it is used, and sometimes that's the only recourse we have. But it's better if you can plan for that heterogeneity beforehand. That's the best chance you're gonna have to increase the power of your study and find treatment effects that are unbiased and with the minimum possible variability. Yeah, does that? Yeah, and really it's kind of interesting because sometimes, you may be very clear that you have something that affects your treatments, in this case, soil type. But sometimes it may not be, right? Sometimes you may not even realize you have variability in your field. And then only after you get yield and you look at your data, it doesn't make sense. Maybe you pull some other soil samples, run some other analysis, and then realize that maybe half of your field was deficient in another nutrient that you did not test for that was important. So that those after the fact known variability sources we can incorporate as covariates in the ANOVA. But if it's better to know beforehand and plan for that in your design. Other questions?
Dr. Bastos,
Yes.
I have one question.
Yes.
In this case, we have taken 4 blocks. What if we have just taken 2 blocks, 1 block for the darker soil and 1 block for the lighter soil, and in each block having 2 repetitions, like completely randomized?
So you're saying basically like if we were to combine block 1 and 2 into one and
Yep,
4 into one as well and have just 2 blocks?
yeah,
So that is a really good question because it's going, like thinking about it is going to impact how many degrees of freedom we allocate to different parts of this ANOVA. We're gonna see a little bit more of that topic in the next lecture here. But so if you think about it, if you have if we have a treatment appearing more than once in a block, which is what, like, if we took block 1 and 2 and merged it into one block, and 3 and 4 and merged it into another block and had 2 blocks, one on each soil type, you would have what is called a generalized randomized complete block design, which is what it means when you have more than one, or each treatment appears more than once per block. What happens is if you have a randomized complete block design like this, one of the problem or one of the limitations of this type of design is that you cannot really estimate, or you should not use the estimate of the effect of the interaction between treatment and block, because you do not have a given treatment in a given block more than once. You just have once. So even though if you, again, this is why this is so important, is that you can run all sorts of wrong models and you're never going to know you run a wrong model. It's going to be up to you to understand that you're running the correct model to your data. So when we're running the ANOVA for this randomized complete block design, if we wanted, we could include in that model the interaction term between treatment and block. We should not include that, but if we do include, you're going to get results back from it. So let's say that in the case that Sekar was saying, where we did have a generalized randomized complete block design, which means you have each treatment appearing more than once per block. In that case, because you do have replication of a treatment within the block, you do have the needed information to test that interaction term of block and treatment, because you do have replication, you do have treatments appearing more than once per block. So what that would give you is more power to say, okay, I do have. So let's say that you run this study here that we're seeing on the screen right now. If you analyze this study, you will likely have a significant effect of the block, because as we're saying, that darker soil is giving you more yield than lighter soil. So you likely have a significant effect of the block. However, you cannot test if there is an interaction between blocking factor, which is soil type, and treatment, with what we're seeing right here on the screen. If you did do what Sekar is saying, which again, just a reminder, is if we were to merge block 1 and 2 into one block and randomize treatments twice within that space without restrictions inside of that block. So imagine that then, let me see, where's my cursor? I don't think it appears here. But yeah, so imagine that now block 1 and 2 are just one block with the same number of plots that we randomize each treatment twice. But it could happen within this larger block that a same treatment falls right next to each other, just like CRD. So in that case, because you do have repetition of all treatments are appearing more than once within the block, you can actually use this information and test to see if the interaction between blocking factor, which is soil type, and treatment is significant or not. If you're doing what we're showing here, you cannot make that test. Meaning that if your blocking factor, so soil type in this case, does interact with your treatment, we cannot test that to see if that's significant. So that effect is going to be compounded inside of a treatment effect. Another way of thinking of this is, let me give another example. So let's say we're doing the design that is on the screen right now, and let's say that our treatment is irrigation. Now, irrigation and soil texture probably interact, right? So if you apply, let's say, 3 levels of irrigation, where let's say no irrigation, exactly what's needed, and then more than what's needed. If you apply that type of irrigation level, maybe the treatment that's needed on the darker soil, applying what's only needed on the darker soil, is gonna yield as much as the over applied treatment on the light soil, because now you're over applying, maybe that water is being lost through leaching down the profile and so on. So you do have this interaction, potential interaction between your treatment and your blocking factor. That can be an issue if you run an RCBD because you cannot estimate that effect. That effect is gonna just be lumped partially into your treatment effect, partially into your block effect. But that interaction is not estimable. It should not be estimable if you're just running an RCBD. However, if you are running a generalized, randomized, complete block design, which is again Sekar's question, you would have two blocks. So a big block in dark soil type, a big block in light color soil type, where each treatment appears twice within each block. Now you have that capacity of actually testing to see if this interaction is significant or not. This interaction between blocking factor, which is soil type, and treatment factor, which was irrigation, the example I gave. And then you can actually pull out that variability that's related to that interaction and remove that from your treatment effects. So again, you regain more of that power to find treatment differences from your treatment effects. Does that make sense?
Yeah, kinda, I understand a bit.
Yeah, I don't think I had not planned for us to really go through that example in code. Yeah, but I can share some resources with you if you're interested, or really if anyone if you're running RCBD, that you have reason to believe that so you're running an RCBD. A lot of times we run RCBDs without really having a strong reason for it. You know, that happens in my team, probably happens in every team. And perhaps the main reason that we run RCBDs is more from a management perspective. So it's easier for me to come here. Let's say if I'm doing, I don't know, if I'm applying something to the field that is not related to my study, that is not related to my treatment, but it's like, I don't know, applying, you know, pesticide or something. If I start applying that on block 1, and then I apply on block 2, and then for some reason I have to stop and come back the next day, it's okay for you to do that as long as you finish a whole block, right? Of course, if possible, you want to do everything the same day. And this can apply to sampling as well. You come here to do soil sampling in your study, and then you sample through block 2, and then starts raining. Not ideal, but it could be, the worst situation would be if you stop halfway through block 2, right? And then you come back after rain. That's the worst, because now if rain had any effect on your whatever you're testing in your soil, you cannot separate that for block 2 anymore. Because let's say you finished block 1 before the rain, block 2, you went through half of it and then started raining, and then you come back the next day. Now the samples before rain may have a different result that interacts with rain than the samples after rain. But because they're in the same block, you cannot tell that effect apart. So it's best for you to either not even start block 2, or maybe go through the end of block 2. And then the next day you come back and do block 3 and 4. In that case, even though rain may have had an effect on the treatment, but at least it's going to be equally distributed, that effect. Now you had half of your treatments that had an effect of the before rain, and the other half had the effect of the after rain, without having unbalance in your data. It will increase your variability, but will still give you unbiased estimations. And so most of the time we do randomized complete block designs, not necessarily because I know I have 2 different soil types, but more because if I run into some of these issues, I can plan to do whatever I'm doing through the end of at least one of the blocks, and then come back after the event, whatever that is. Right?
I
But
have a question.
yeah.
So, see, RCPD is like good for everything that is related to soil, or like what things have to do with like fertilizers. But what if we want to study some atmospheric carbon sequestration levels, like we are not considering soil type in that case? So then CRD will be good, or like even then we have to go for RCP, like.
So the question that you should ask is, do I have homogeneous experimental material as far as that impacts my response variable, right? So if my response variable is yield, then I should be thinking, you know, is this variability in soil type important for yield? Because I mean, we're gonna have variability in all sorts of things all the time. That's just nature, right? It's more about thinking if whatever you think there is variability there, if that's impacting your outcome. So in your case, so if you're measuring soil carbon sequestration, is that?
Atmospheric carbon, like how much carbon dioxide it can fix, leaves can fix.
Okay. So then if that's your response variable, what you would be thinking is there anything that I know about this field or this greenhouse or whatever it is that I know impacts your response variable and yet is not homogeneous in my experimental material? You know, so I mean, I'm just trying to come up with something here, but I'm sure you can think of better cases for your own research. But let's say if you know that, you know, I mean, soil type perhaps is this big umbrella because different soil types can have multiple things that are impacting yield, right? It could be texture itself, it could be water retention, it could be nutrient retention, it could be pH. There's a lot of things that kind of go underneath soil type. But in your case, let's say that maybe it's not even a soil type difference, but maybe there's part of your field that has more potassium than others. And then maybe the parts that have more potassium are providing enough potassium for the plants to perform well and therefore fix carbon, whereas the parts that have low potassium levels is below the threshold that would be optimal for the plants. So the plants under there may be undergoing potassium stress, meaning that they're going to be fixing less carbon, right? So that would be an example that in your case, that, I mean, and again, it doesn't have to be related to, it could be related, for example, with nematode. Let's think about something else outside of fertility. So if you have pockets of your field that you have a higher nematode infestation, that will for sure impact your, I mean, most likely will impact your crop response of any sort. That could also be a blocking factor. Does that make sense?
So I asked that question because for my experimental fields, I am using CRD, completely randomized design. So that's why I asked like why we are using that, because we are not studying any soil effect on plants. Like we are just going for poly crosses. Like I am working with switchgrass, so it is outcrossing. So I am trying to get progeny with higher yield.
Okay. Yeah, I guess, you know, again, the framework of thinking of this is, are you aware of any source of experimental material heterogeneity that can impact your response variable? That's kind of the question you come back to, and then you think from there, right? If you think there is, then your study is probably not going to be as powerful as what it could. So by powerful, I mean you're not going to have enough, or not going to have as much power to find treatment differences as you would if you had done a different design. But it is very possible as well that, so it happened to me a lot, and if you've been running your analysis, you may have seen this, where you do an RCBD, either because you know there is an issue, there's variability, or maybe because you just want to be on the safe side in case you need to abort sampling or something like that. And then when you're running your analysis, block comes highly non-significant. It's like p-value is like 0.9. What that tells you is, at least for that year, that site, you could have potentially used completely randomized design and you would have been fine. In fact, it's an interesting topic because if your field is homogeneous and you run a randomized complete block design, you're actually losing a little bit of power because you're consuming degrees of freedom to estimate the block effect. Whereas if you don't have a block, if you have a completely randomized design, you're not consuming those degrees of freedom, which means that they go to the error. And the more degrees of freedom in the error we have, the more power we have in the analysis. So it's like trade-offs, right? But let's say if you run a randomized complete block design and it comes back highly non-significant, maybe next year you can plan for a CRD on that area. Or maybe after that whole study ends, because you do want to keep your same design throughout that study. But then, you know, in future projects in that same area, similar to what you're doing now, then you could run as a CRD potentially. Good questions, great discussions. Other questions? So let me just do a show of hands here. If you're not running, if your research does not relate to CRD or CBD, you don't have to answer, but if your studies are running CRD, can you please raise your hand, just so I see a show of hands? Okay, so just to see Chris. Okay, and then RCBDs. Okay, so most people, I guess, are running RCBDs. Yeah, so that would be, yeah, those, all these questions and conversations are really useful, I guess, to kind of understand when to use and what happens when you use one or the other, especially if you use them in the wrong situation. So as we were saying, yes,
Excuse me, Dr. Bash, just like in that carbon sequestration question, like can the response variable be temperature, vapor pressure, deficit? Because those are the atmospheric parameters that affect the sequestration of carbon as well. So can we use that as a response variable?
so the response variable is what would be on the left-hand side of your formula, right? So
Yes.
let's say if you're explaining, if your response variable is carbon sequestration,
As a
then
function of
temperature would have to
temperature.
be
Uh-huh.
an explanatory variable, right? So it'd be on your right-hand side of the formula.
Mhm.
So you could use as covariates, but then you would have to be measuring those for each plot at least, right? Because let's say if you use weather data for the whole field, I don't know how useful that would be really, because now we're talking about changes of carbon sequestration on a plot basis, but then comparing everything with one value for temperature, let's say, right? However, if you do have that covariate data at the plot level, you could potentially use this as a covariate on the analysis, if that's the case.
So I might not have gotten that he is calculating, like he's observing the data from the plot basis. I'm sorry.
Yeah, no worries. Awesome, thanks, Kriti. Any other questions here before we move on? So I think, so today we have another lecture that I think is really important to set the stage for how we're going to analyze this data. And then probably next week, I'm going to send you a data set. We're going to be doing these hands-on. So we're going to see how we analyze it, how we make sure we're doing the right thing, and how we address some potential issues that may come up. All right, thanks for the questions. I enjoyed the discussion. Anyone else either has a question or maybe something you'd like to share from your own research, your own experience? So let me pick on the control environment students for a moment here. So for you, what would be a common blocking factor for your?
So I'm working right now within the greenhouse. So usually we have different conditions within the same greenhouse. The entrance then on the other side of the greenhouse. And in my case, I was blocking by bench. Then in the same bench, I will include different trays with each treatment within each tray. So it would be bench block, and then each tray would be my replication with
Mhm.
adding my treatments. And then the observational units, how many plants I have within each tray.
Nice.
That was ideal, but I didn't do that.
Yeah, okay.
And that was an issue actually in the research, because I'm doing exactly what you told us not to do.
Okay.
And it's considering your observational units as cell replicates.
Okay,
My limitation is that I had just two trays instead of three because of the size of my trays. And we try to find a way around using those cellular replicates. But now I have a question actually, since the experiment is more towards prediction rather than differentiating between treatments, how significant would be to redefine my experimental design if my objective is more towards prediction rather than treatments?
Okay, so let me just repeat what you said so people online can know, because it was a really great point. So Jonathan was saying, so Jonathan works in controlled environment agriculture, so inside of a greenhouse, and he was saying that it it would be common, I guess, that in that case you have, because with inside the greenhouse, maybe the air outlet is in one side. So either if you're cooling or heating or whatever you're doing with air is going to have a gradient on your study, right? So imagine the same gradient as, let's say, temperature in that case would be our soil types here. So it would ideally would have benches that would be your blocks that would be parallel, no, perpendicular. Yes, perpendicular to your gradient of temperature. Could be the same thing as we think about elevation or soil type in this case. So he was saying that that would be a common design, even though they did the opposite. And then he was asking if that has an impact in his case, since he's using that data mostly for prediction rather than comparing means, what we do when we run ANOVAs, right? So one of the reasons why well-designed experiments are still, like, this is something that has been developed a long time ago, right? So if you I don't know if you are familiar a little bit with the history, but a lot of this was developed either by or in collaboration with Sir Ronald Fisher when he was working at Rothamsted, which is one of the oldest experimental institutions in ag, it is the oldest in the world. It is in the UK, where he basically got there. There was a lot of data that they were already conducting studies on, and he just had to figure stuff out. And he was a statistician, so he developed a lot of the things we use today. And this happened, I don't even know when, but it was a long time ago. And we're still using the same models, same designs, right? The reason for that is because they're very robust if you run them properly, as we're talking about here. And the reason for that, the main reason there is if you are randomizing, so randomizing is a very key concept here, because when we do randomization, we are allowing us to make this assumption of independence, which is big here. So if you don't randomize, let's say you just have treatments 1, 2, 3, 4, 5, 6, 7, 9, all the way down, and then again 1, 2, 3, 4, 5. That is not randomization, right? That is just you assigning treatments in basically numerical order, which has a bias, even if it's not explicit bias, but it has a bias. So the fact that we randomize is what allow us to treat these experimental units and the data from them as independent, which is one of the big assumptions of analysis of variance. So randomization, especially randomization in homogeneous experimental material, right? So those two things together are what kind of clears the noise in ag research. Because I'm sure working in ag research, we have so many potential confounding effects everywhere, right? This is nature. Nature is so random, or maybe not random, but I mean, also random, but there's so many interactions at play that we need to find good ways to control for all those and really see the signal from your data. So doing randomization in homogeneous experimental material allows us that. In your case, you probably did have replicate or randomization, but you randomize like within the gradient, not like, so your same bench had multiple potential differences in temperature from that air vent. What that does is it's going to add noise to your data. And in this case, because it is more of a machine learning approach, where in machine learning we don't normally directly assume independence. We just do observational data that has all sorts of potential correlations and confounding effects inside the data. And we really rely more on volume of data than trying to model the covariance between those data points, right? So in that case, as long as you have a lot of data, and I guess a lot of data can be very subjective, you would still be able to create a model. Now, let's think about it. So you have all this data. Let's say you have all this data that has all sorts of confounding effects because it was not properly randomized, but you're using it for machine learning, right? In this case, you're still going to be able to create a model, and within the model that you're using, different algorithms may or may not consider that correlation between your observations and how they relate to what's going on in the environment that they're in. And let's say you have a model that you train and you get a prediction R square of, let's say, 0.6, right? Now, maybe if you had trained the same model collecting data that were properly randomized and assigned, maybe the R square could be higher. And maybe it is not higher because there's some effects that are coming through your response variable that you cannot separate them. You cannot fully say, oh, this effect here is because it was higher temperature, whereas this effect here is because it was more nutrients. They're kind of combined, right? So it's harder to separate those two. So it will potentially create machine learning models with less predictive power than they could have if you had used observations that were fully independent and properly randomized.
So professor, so in this case, can't he add some another covariate like the temperature gradient within the greenhouse and then put that into a machine learning model? Will that increase the R square?
It could, right? So it could, if you do have a covariate that is causing that confounding effect and you can add it to the model, it could help. The thing is that, we are assuming that temperature is the main factor there, but there could be other things that perhaps are correlated with temperature, but are not temperature necessarily. So maybe is vapor pressure deficit, or maybe is the speed of the wind that comes and dries the leaves more in that side. So, I mean, we're kind of simplifying that problem to say temperature is the problem, so let's just model that. And it could work to some extent. But at the same time, maybe there are many other variables that could also help you to make better models to account for this confounding that we're not even thinking, right? Or that maybe we didn't measure. So partially we could, but the best way would really to do this in a way that it was, not confounded, I guess.
In fact, that was one idea, like trying to get sensors that are trace where you can like add more like granularity to the temperature you are gathering from each part of the ring. Challenge was we want to establish like a more general model without considering the treatment per se.
Right.
You just take like what you're getting from a camera, and that's the only predictors that you use, and see how you can generalize that. But without adding so much like environmental information that of course it's going to change over time.
Yeah. And also another thing here that we're going to talk in the next lecture is cause and effect or just correlation by itself, right? So normally with machine learning types of approaches, we're really relying on certain correlations, but it cannot directly say there is a cause and effect, right? Because you did not control for that, in a way that would keep the other confounding factors to a minimum, and you can really see that cause and effect, which in design experiments like we're seeing here, we can directly associate cause and effect because of randomization that being done properly on homogeneous experimental material, right? I can really say that my treatments are what is increasing soil, or my treatment is what is actually increasing yield, and not a combination of treatment and soil type that is mixed in the signal that I cannot dissect if we use the proper design, right? So in your case, you may still be able to create a model that improves if you give those covariates that are correlated with your response variable. But it's going to be, normally we don't associate this strong cause and effect coming from machine learning models. We more have an idea of what's impacting, and then if we really want to say this causes that, then we take that, what we learn from there, and then we run a study where we control those effects and randomize properly, right? Yeah, so really another difference here between, I guess, machine learning and experimental design is with properly designed studies, we can directly derive this cause and effect relationship. That is not something that we can or perhaps should do when using machine learning models. All right, great questions and points and conversations. Anyone else would like to add to the conversation? Okay, I don't hear anything, so I'm gonna move on to our next lecture. So going back to the website, there is this slide deck here for today's class. If you open that, I think on the title at least, it may look very similar to the one we just saw, but it's going to be more focused on completely randomized design. Okay, so with this talk here, I do want to talk a little bit more about completely randomized design. I want to lay some of the foundation of what we're going to be doing in code. So we're not going to be coding quite yet, but I did want to show you visually some of the concepts of what we will be doing in code, just so you understand better and not simply see code in front of you. Okay, so here, well, let me do this. Maybe we can take a 5-minute break in case you need to go to the restroom, and then we'll come back at 10:53. Yeah, let's do that.
When you finish Dr. Astor's presentation today,
I finish.
will you? Will you plan to finish?
Oh, this presentation? Maybe, I guess. I mean, depends on,
30 seconds per slide.
how many discussions we have. So probably not.
Just today class ends at 11:10.
No. Oh, is it?
I think so.
Yeah, I need to watch those more closely.
Going good now.
Yeah, that's right.
On Thursday, it's 11:30.
Yeah.
That's when you give a break at the lunch.
Yeah, that's right. So probably not.
No.
All right, so, yeah, I keep thinking that we have until 11:30 both days. And Amrinder just reminded me, we don't have until 11:30 today. So to make sure that I end up on time and you can, you don't have to run behind if you have anything after this class, we'll make sure to just take another 15 minutes here and get started on this lecture. Okay, so, as we talked about, we're gonna be, I guess, going through the workflow of two different experimental designs. CRD is the first one of them, so let's get into that. So the goals of this lecture here are to talk again about homogeneous experimental unit, treatment randomization, the effects model, the ANOVA table, and the linear model assumptions. So I do imagine and hope that you have already seen some of these. Perhaps I'm going to be showing you a slightly different angle or take on these, especially applied to some data that may seem more familiar to you. If you have not seen these, I think it's going to be a good background. If you have seen, maybe you also gain something extra from this, looking at a different angle. All right, so, we're going to be referring to our motivational example here, where we have the one we just talked about on the previous lecture, where we have a 2A factorial with nitrogen fertilizer rates, 0, 100, and 200 kilos per hectare, and then our potassium rates of 0, 30, and 60 kilos per hectare. So we arrived at the conclusion that for that, if we wanted to explore both the main effects, but also the interaction, we needed to have all treatment combinations, which give us 9 treatments total. And assuming that we have homogeneous experimental material, so same soil type, topography, temperature gradient, or no temperature gradients, or anything like that. So we have homogeneity in our experimental material. We could use a completely randomized design. So, we're going to be talking about some of the points that we just had a conversation about, that randomization is what guards us against unknown or uncontrollable sources of bias. It helps us avoid systematic patterns and eliminate selective assignment of experimental unit to treatment, either conscious or unconscious. And this is really important, a really key concept, because again, let's say I see I remember when I was doing my undergrad and I was taking an applied stats course, design course, and we were on the field. And, to me, it seems like, well, I could simply have my first block being 1, 2 treatments, 1, 2, 3, 4, 5, 6, 7, 8, 9, and then start randomizing on the second block. And then I remember my professor saying, well, I mean, yeah, you could do that potentially, but you're not randomizing fully that first rep. You're just following the order of your treatments, which probably have some meaning to you. Maybe you selected treatment 1 because it was a check, and then treatment 2 because it was a low rate, medium, high, and so on. So even though you may think there is no bias, there is probably bias around whenever we do not randomize, right? So we want to avoid that bias, even if that is a conscious or unconscious bias. And really, randomization here is what allows us for valid inference on causation. If we can randomize on homogeneous experimental material, that allows you to say, I changed this and that had an effect on this. I changed nitrogen rates and that had an effect on yield, rather than just having correlation. So because we have homogeneous experimental material in this example, then randomization of any treatment to an experimental unit is unrestricted. We already talked about this, but it basically means that 2 replications of the same treatment could fall into neighboring plots or pots or whatever it is on your case. In our case, plots on the field. So in our motivational example, we had 4 replicates for a total of 36 experimental units. In our case, experimental unit means a plot on the field, which is something that we just saw. So I'm not going to spend a lot of time here, but because we have homogeneous experimental material, in this example, soil type, We can use a CRD, and it does not matter if the same treatment falls every single column of this field, because it is all homogeneous, and because it was randomly assigned, we are safe against, or we are making sure that our design allows us to estimate treatment means that are unbiased and the most stable or the least variable possible. This is just those notes on treatment 1 appearing 2 times in one of the columns and 0 times in another column. And because we're using the correct design for this case, we should be good. Okay, let's talk about the effects model. We're not going to spend too much time on statistical notation like this, just when really needed. And this case here is really needed because it directly connects to your ANOVA table, which directly connects to your model when you specify it. So this is the effects model for, well, let's just go through it. So YIJK is the observation on the kth replicate, or in this case block, from the ith nitrogen rate and jth K rate. So this IJK is just an observation that you made on your field. That is what you're actually measuring. That's your response variable. To create a model that explains that observation, we need to have these components here for the design that we're talking about in our motivational example, where we have mu here is the overall mean. Alpha I is the differential effect of the ith nitrogen rate. So imagine that we're going to have a coefficient for each one of your nitrogen rates. We're going to have an alpha coefficient for each one of your nitrogen rates. Then beta J is the differential effect of the jth potassium rate. So we have 3 potassium rates, so J equals to 3. We're going to have 3 beta coefficients, 1 for each one of the potassium rates. So overall mean, main effect of nitrogen, main effect of potassium. This is the interaction term. So this would be alpha beta of IJ, which is basically the differential effect of the combination of the ith nitrogen rate and jth K rate. So this would be your coefficients for the interaction between nitrogen and potassium. And then finally, our error or residual term. So imagine that we're taking these pieces and I'm saying, this observation I have here, I can get to, or I can get close to it, or actually I can get to it by knowing how much of its value is coming from the overall mean, how much of its value is coming from the main effect of nitrogen, main effect of potassium, their interaction. Then still there's going to be uncertainty, right? Because we're not going to be able to explain or predict something 100%. So there's always going to be error or noise that is left after you account for all of your treatment and experimental design factors. In this case here, it is a completely randomized design. So we have so really the effect of the design in this case is ending up in the error because we have no other factor in this design that is influencing randomization. So the design is really, the design effect is ending up in the error. And the error is what is left of that observation after you account for all those effects in your model. Okay. Now, let's look at an ANOVA table here, and there's going to be, well, I guess the next slide here. So this is just, whenever you're studying, going back to the slides, it's just some notation of what you're going to see on the ANOVA table, but lowercase n is the number of levels in nitrogen rate, which in our case is 3 levels. Lowercase k is number of levels in potassium rate, which in our case is also 3 levels. Lowercase r is the number of replicates. In our example is 4 replicates. And uppercase n is the total number of observations or experimental units, which is basically our treatment. So 3n, 3k, and 4 reps. So 36 experimental units. All right, I think this is probably a good place for us to start and hopefully finish in the next 9 minutes here. Let's really go through what is going on here, because this is really an important concept, especially the more complicated design you have. This is going to be really important. Okay, so let me just have some idea of your familiarity with this ANOVA table. If you understand what's going on here, at least 80% of what's going on here, please raise your hand. Okay, so we have maybe half of the class that knows what's going on here. That's great. But you know, another part of the class does not know what's going on here. So let's take this slowly because it's going to be very important. Every ANOVA table, and this is something that you may think, okay, it's just an ANOVA table. Why spend so much time here? Is because looking at your ANOVA table is going to be the only way that you have to check if your model matches your actual design. And we do that mostly through the degrees of freedom column. But the ANOVA table is what I always use to make sure that I'm specifying the correct model for that data that was collected based on a design that has a model, right? So if I make a mistake, if I'm expecting to have a certain number of degrees of freedom and I don't see them, it means my model is not correct. And this ANOVA table here, you can and you probably should do this, at least like the sources of variation and degrees of freedom. You could and should do this for your research even before you collect data. Just so you know that whenever you need to specify this model, you already know what to expect on the ANOVA table when you see it. Okay, so let's walk through what's going on here. Normally, you would see ANOVA tables that at least have a source of variation and potentially like a p-value column, which we don't even have here. But source of variation is important because this is basically going to have a line for each one of your of the terms on your effects model. Okay, so if you look here, there's N rate, potassium rate, the interaction, and the error. The error is the residual, is that E term. If I come back here to my effects model, well, it does have the overall mean that I'm leaving out of the ANOVA, but everything else is here. So the effect of nitrogen, effect of potassium, their interaction, and the error. Knowing your model also helps you to at least understand that you're getting the right ANOVA back after you run your model. Okay, so main effect of nitrogen, main effect of potassium, their interaction, and the error. First thing is you need to know your source of variation, right? If this was an RCBD, it would, even the same treatment design, if this was an RCBD, it would be a different effects model. It would be a different ANOVA table that we're going to see when we get there. But just so you know, the ANOVA table is what's going to give you hints if you have the right model, if you're thinking the right direction of what you actually designed the study to be. Then we look at degrees of freedom. These degrees of freedom here, the way they're calculated is basically, if you remember, n in lowercase n in this case is the number of levels in nitrogen. So 3 minus 1, this is the formula. We apply to 3 minus 1, we get 2 degrees of freedom for nitrogen. Same thing for potassium. Whenever we had interactions, it's just, this is the formula here. So we do this calculation here for one of the factors times for the other. So in this case, we would have 4 degrees of freedom in our interaction. And then the error follows this formula here where N and K are 3 and 3. R is 4 because the number of reps, and we end up with 27. So here's the key as well. If you are running the wrong design, it may be, or maybe you're running subsampling as true experimental units without realizing. You go out to your field, you know you have 36 plots, which means you should have a total of 30. Well, in this case, a total N is 36. We do -1 for that effect of the overall mean. We should have here 35 degrees of freedom. You can think of each observation coming from an experimental unit. So each true replicate gives you 1 degree of freedom of information to spend on this ANOVA table. So that number should match here in the end. If you run your ANOVA and your error degrees of freedom comes 3 times higher, you are running the wrong model because you are not properly modeling or accounting for subsampling. What happens is if you're running the wrong model, you're going to have, well, I guess let me hold this comment for when we get towards the end of this ANOVA table here. But anyways, checking the number of degrees of freedom of your error term is going to be really important to check you're running the right model. Okay. So degrees of freedom, very important to determine properly. Our sum of squares column. So this, it has a whole formula that's basically taking differences from your observations to different levels of your ANOVA. We're not going to really go through that formula here. You've probably seen this already in a stats course. If you have not, you can go back and Google for it. But I just wanted to, you can kind of think of your sum of squares as a measure of how much of your total variability is being explained by that factor. So the sum of the squares of nitrogen is how much of my total variability is being explained by nitrogen itself. And then I have one for potassium, one for the interaction, and also one for the air. Ideally, what we want is to have a lot of this variability in the data to be explained by our treatments, and very little of it to be explained by air. Because that means you're really associating most of the variability with what you're really interested in, which are your treatments, and very little of it is being extra noise that ends up in the air, right? So the more noise you have, the more difficult it will be to find differences. The less noise you have, and therefore the less signal you have, so variability being explained by the treatments, the easier it was going to be to find treatment differences. So that's the sum of the squares. The mean squares column here is basically just a ratio for each one of these rows here, the ratio of the sum of the squares by the degrees of freedom of that column. So we just do that for each one of these rows. So the main effects of N and K, the interaction, and also the error. And the F ratio finally here is what we're gonna use to then calculate p-values for each one of these rows of this ANOVA table. So if we see here, I want you to pay attention to this. So the F ratio for the main effect of nitrogen is going to be the mean square of nitrogen, which was just calculated here using the sum of the squares and the degrees of freedom of nitrogen, divided by the mean squares of the air. The same is for potassium. So it changes the potassium part, but is divided by the mean square of the air. Same for the interaction being divided by the mean square of the air. What this is doing, and what I want you to get out of this, is that it is using the mean square of the air to test the effect of each one of these actual effects that you're interested in, right? So we're interested in nitrogen, potassium, their interaction. And I'm using the mean square of the error to test for them by doing this ratio with each one of them. And then this here comes back as a value. So the higher is this value, the more evidence you have that that treatment effect is significant. And I think I have a few of those sentences here. Yeah, so each component of your effects model ends up being a row in your ANOVA table. And the larger is this ratio here, each one of these three, the more evidence you have that that factor is significant on your ANOVA. Okay, I think we can stop here. Next class, we're going to talk more about this ANOVA table, really make sure you understand what's going on, understand some of the trade-offs here, and then talk about CRD. And I will probably introduce the coding exercise for how do you actually do randomization for a CRD in R, how do you create your plot layout, how do you run a model, and check for assumptions, and so on. So we're going to do that next class. Let me open here. Let me open here the attendance. All right, make sure you claim your attendance, and then we should be good after that. Thanks, everyone, and I'll see you on Thursday.